Hello, grade 11. How are you today? This is week 6, and we are going to talk about pedigree. Pedigree is a method of investigation used by biologists in order to study the transmission of traits and diseases in a family. How are pedigrees used to study human traits? In pedigree analysis, a series of symbols are used to represent different aspects of a pedigree. In the adjacent document, we have the square for the male, the circle for the female. When the square or the circle is colored, it means that the individual is either affected or carrying the trait. This line joining the square to the circle means that there is mating or marriage. This is the offspring in birth order. The number of generation is represented in Roman number, while the number of individual is the normal numbers. This is for identical twins and these for the fraternal twins. Modes of transmission of a trait or a disease. First, we'll start with the autosomal inheritance, autosomal recessive inheritance. As you see in the adjacent pedigree, two phenotypically healthy parents gave birth to a diseased child, example 2-2. Then this means that the trait under study is recessive. In this case, most affected children have unaffected parents. Heterozygous have an affected phenotype. Two affected parents will always have affected children. Affected individuals with homozygous unaffected mates will always have unaffected children. Close relatives who reproduce are more likely to have affected children since the gene pool is becoming smaller. Both males and females are affected with equal frequency. The trait tends to skip generations. When both parents are heterozygous, approximately 1 over 4 or 25% of the offspring will be affected. As an example, we have albinism and diabetes. The second mode of inheritance is autosomal dominant inheritance. As you see in the adjacent pedigree, we have affected individuals have at least one of their parents affected. The trait does not skip generations. So as conclusions, we have affected children usually have an affected parent. Heterozygotes are affected. The trait does not skip generations. Two affected parents can produce an affected child. Two unaffected parents will not have affected children. Both males and females are affected with equal frequency. Example, polydactyly or having extra fingers or toes, and Huntington chorea, which is a neurologic disease. As an application, we can see in the adjacent pedigree, this is albinism, which is autosomal. We have, as an example, this couple, 10 and 11, which are phenotypically healthy and gave birth to a diseased child, let's say, example, for 13 or for 15 or 16. So this means that the trait under study is recessive. So let's start with the questions. Identify if the allele of the disease dominant or recessive. Since two phenotypically healthy parents give birth to an albino child, then the allele responsible for albinism is recessive and masked by the normal allele, which is dominant. Second, if you want to designate by symbols the corresponding alleles, we can choose capital N for the normal dominant allele and small a for the recessive albino allele. 3. Specify the genotypes of the individuals 1 and 5. Individual 1 is diseased and the disease is recessive. This means that in order to show this trait, it has to be present in two copies, A. So the genotype of individual 1 is small a, he is albino, and the allele responsible for albinism is recessive, therefore it's not expressed unless it is in the pure or homozygous state. Second, individual 5, it's NA. Since individual 5 is healthy, then he must have the normal allele capital N. But since his father is diseased, having the genotype small a, he must inherit this allele from his father. So the alleles N and A represent his genotype. And finally, I would like to thank you. Have a great day.